Random mating, an assumption of Hardy-Weinberg equilibrium that the alleles are able to mix in basically a large gene pool without regard to what other alleles are circulating in there. Well, let's violate that assumption of Hardy-Weinberg and see what happens when organisms get to pick their partners. In this first part of the lecture, I want you to be able to define inbreeding, inbreeding depression, and your extinction vortex. Be able to calculate inbreeding coefficients and calculate inbreeding depression. Let's get going. So, mating is often not random. In fact, many organisms are going to choose their mates. If you were to consider the sounds of spring, where you have beautiful bird calls and insects fluttering about, and of course, flowers displaying their beautiful petals, it's all really a lot of organisms that desperately want a mate and are trying to pick the best mate out of all of them. Random mating is really considering mating random as according to a certain allele. So let's say the allele for leaf pigmentation gene number 43C in a certain type of poppy. As long as the organisms are mating, or these poppies are mating without regard to that one gene, it is random, effectively, even if the poppies that are yellow are mating with only other yellow poppies, and the orange poppies mating with only orange poppies. When we're considering a different allele that is random in the population, then that is random mating. Choices, however, can be made a couple of different ways. Organisms can choose to mate with the organism that is closest to them. I mean, heck, if you're planted in the ground, you can't really move and find a different mate. Choices can also be on self-recognition, where an organism will mate on other organisms that are most like it, thus assuring that it's mating with the right species, however, increasing the probability that it's going to be inbreeding. It can be choices made on attractiveness, and we're going to circle back to that in the second part of this three-part lecture. And of course, it can be measured in genetic incompatibility. So, mating systems are going to help determine the outcrossing of different organisms. So, for mating choice, let's think about different mating systems. Some organisms are going to be able to choose their mates. Here you go, I have a cat here. Some organisms are just going to broadcast their gametes around. So, whereas cats, there you go, can pick their mates based on a mating display and such, other organisms such as grasses are just going to broadcast alleles. For corals, they're going to just broadcast their sperm into the water, or their eggs into the water at a certain time of year. For grasses, they broadcast their pollen out into the air at a certain time of year, and thus any mating choice has to be done at the post-reproduction moment. When the pollen has already landed on the stigma of a plant, it can then pick whether or not that pollen will germinate or fertilize. But that's a very genetic component and definitely not an active mating choice. There's also some organisms that are going to broadcast their sperm and eggs, but be close to their chosen mate at the time. This would be an example of salmon, where a female salmon will build a redd, which is R-E-D-D, it's a gravel pit, and will lay her eggs in the redd. And the male salmon that she has chosen to mate with her will come alongside and broadcast their sperm directly upon those eggs. Whereas a sneaky salmon may come upstream of them and kind of just spray some sperm down into the eggs in the hope that some fertilization will occur. So this ensures that mating is kind of chosen, but at the same time there is an element of randomness. Herring are another example where the female herring will lay their eggs on grasses and the male herring will just spray their sperm into the water near those eggs. However, the waters become thick with sperm and even look a different color, so you know that there is some randomization going on there. And then we have terrestrial vertebrates, which have internal fertilization, where a hamster will pick only the hamster it wants to mate with and will stay with that other hamster for a certain amount of time. And this kind of internal fertilization is a more extreme method of mating choice, which we will circle back to through sexual selection. Some choices are just to mate with what is closest, and that is going to lead to inbreeding. Organisms with low mobility frequently stay close to the parental ranges. The acorn doesn't fall far from the tree, and since the acorn doesn't fall far from the tree, it is likely that when that acorn grows up, becomes a tree, and creates female flowers, it will receive pollen from its parents and siblings. That's a bit of a problem. and that can lead to inbreeding depression. Inbreeding itself is mating with genetic relatives. Look at this cat. It's inbred. Haha. Okay, moving on. I just love that cat. Anyway, there are certain mechanisms by which organisms that are passive in what pollen they receive or sperm they receive can actually ensure that they do not get fertilized, but that's for a different class. So this is a time to think about why inbreeding might be a taboo in many cultures. Many human cultures are going to say that inbreeding is simply not acceptable, that you cannot marry your sister or brother, and you can actually find biblical laws saying that inbreeding would not be allowed. Consider what might actually cause that. And at the same time, take into consideration how many times you actually see inbreeding in these same cultures and why it is justified. For example, you'll see that when some of the biblical patriarchs would look for a spouse, they would look at cousins or second cousins because that would ensure genetic purity. So think about some cultures like the ancient Egyptian pharaohs where inbreeding was enforced, in fact, or for perhaps the royal blood examples where certain traits like the connection to the divine mandate, however you may measure that, are going to be only available to the blood and those of the blood. Or House Lannister, which is just gross. Anyway, you can see here the ancestry of King Charles II of Spain and how a large amount of inbreeding occurred with the Habsburgs. Now, of course, you may also remember the dynastic relationships of Habsburgs to other various royal houses, such as the Tudors, well, not the Tudors, they definitely didn't want that, once, didn't end well, and the Bourbons and all of that, where the Habsburgs wanted to maintain their claims to the certain lands that they had secure through marriage and intermarriage, which it was a ridiculous way of doing things that ended up with Charles II of Spain. Look him up. You're in for a treat. A drooling treat. Anyway, royal blood is one of these examples of extreme inbreeding enforced and codified by certain chivalric ideals. But what does it lead to? Well, let's talk about genetics and the genetics of inbreeding. One thing it's going to lead to is homozygosity. So let's say we had a bunch of heterozygotes. We're going to start with heterozygous organisms, but homozygotes will only mate with homozygotes. So that is already in a type of assortative mating. If homozygotes specifically mate with homozygotes, or let's just say all these organisms mate with self or very close relatives, okay? Homozygotes mating with themselves or mating with other close relatives will beget more homozygotes. Just remember your Punnett square, of course. Two heterozygotes are going to make a homozygote, and it will make as many heterozygotes as homozygotes, but homozygotes, of course, can only make more homozygotes. So homozygotes making more homozygotes will lead to a lack of genetic diversity and a loss of overall heterozygosity in the population. It's a speedy loss of genetic diversity and will enhance genetic fixation. All right. So let's look at these genotype frequencies in terms of the coefficient of inbreeding. Increasing the number of homozygotes doesn't actually change the allele frequencies. It just changes how the genotype frequencies are arranged in the population. An excess of homozygotes in a population is actually a good way of determining that in the past there may have been inbreeding, such as may have occurred during a genetic bottleneck or founder effect. This can, as I mentioned earlier, lead to the enhanced fixation of alleles. See a previous lecture. And we can calculate how much inbreeding there is using the coefficient of inbreeding. This is going to be a direct measure of how inbred a population is. And it's called F. So that's the probability that two alleles are identical by descent. So they're coming from the same parent. A probability that the alleles are not identical is going to be 1 minus F. That's the probability that these alleles came from different parents. So if we wanted to look at the frequency of AA individuals, homozygous dominant, we're going to have P squared, which is your frequency of the A, uppercase A allele, multiplied by 1 minus F, plus P, the frequency again, times F. That's the frequency of your AA individuals, given that these are not inbreeding. Your 2PQ times 1 minus F is your frequency of the heterozygote individuals. Well, what's F? How do we calculate F? So let's say you have a grandmother here that has 2 offspring, a mother and a father for the focal female. So for this focal female, the copies of an allele came from grandmother and they came through the parents. So what happens here is you have the 2 offspring, the 2 eggs from the grandmother are 50% related. They're 1/2 of the same alleles. The sperm from the father is 50% related to the egg from the grandmother. That's through the products of meiosis. It's got 50% of the genetics from the grandfather, and this is kind of an on average thing. Go back and review meiosis if you want to get the full why. But the sperm from the father is 50% related to the egg from the grandmother. The egg from the mother is going to be 50% related to the egg from the grandmother, because there is grandfather DNA there too. So what's F for that focal female who is the daughter of this mother-father bond, the brother, sister, mother, father kind of thing? That'd be 1/2 times 1/2 times 1/2, or 1/8. What we see here is also, we can see for this focal female, that's 1/8. For full sibs, so this is, these individuals come from the same parents, so it's 2 parents, son and daughter, have a focal female here. It's actually going to end up being 1/4 because we're looking at 2 loops of that 1/8, where the sperm from the father is 50% related to this egg from the grandmother, the 2 eggs from the grandmother are 50% related to each other, and the egg from the mother is 50% related to the egg from the grandmother. Okay, so that's 1/8 right there, the 1/2 times 1/2 times 1/2. From the sperm, though, or from the other side, you have the egg from the mother is 50% related to the sperm from the grandfather, and the sperm from the grandfather is 50% related to the other sperm from the grandfather, and the sperm from the father is 50% related to the sperm from the grandfather. So tracing it patrilinearly, you're going to see a 1/8 as well, because you have these full loops. So if you see with the grandmother with 2 unrelated grandfathers, in A, you have an 1/8, an F of 1/8. For 2 parents with 2 siblings with 1 daughter, you're going to have 1/4. And it's a complex way of calculating F. I don't know if any of you are people who have read Flowers in the Attic, but it is basically dealing with exactly this and the drama around it. But it is a book series by V.C. Andrews in which you have 4 children who are abandoned by their parents and left to live with their grandmother. Now their grandmother doesn't like them, so she locks them in the attic, and eventually, spoiler alert, poisons a few of them with arsenic. Okay. However, the eldest daughter and the eldest son have their own offspring, which is going to lead to sequel after sequel. What we see here is effectively, and they find out, that's right, the grandmother locked them away because they're sinful children, because they're all offspring of a mother, father they're both children of the grandmother. So we can see this actually go out through several lines where A is actually, the focal female from A is the mother in B. So that's confusing enough. But basically that's going to be calculate how genetically related they are. And that will also make you able to calculate the homozygosity. Why is it a bad thing? Well, inbreeding depression. So over time, an increase in the number of homozygous individuals is actually going to lead to inbreeding depression when those individuals are homozygous for a deleterious allele. And there are, of course, populations of humans where they wanted to keep the blood pure, where you're going to see inbreeding depression here. So here we see mortality rate among children of unrelated parents and first cousins. Now, I want to point out that in the American culture, marrying a cousin is not considered taboo. Once. If you have marriage of cousins frequently, however, you're going to see an increase in mortality. It's not a taboo in the American culture. In some cultures it is, in some cultures it's encouraged, whatever. Cultures be cultures, and each one of these data points is a different nation. what we see is a gray line, which if the mortality rate among children of unrelated parents was equal to that of children of first cousins, this gray line would be a perfect fit for the data. What you see, however, is almost all of these data points, yes, there are two that are below the line, but the rest of them are above the line, which means for almost all cases, the mortality rate of children of first cousins is higher than the mortality rate of children of unrelated parents. And you're going to be measuring inbreeding depression in mortality, or reduced fecundity, to be fair, which is effectively mortality of younger. So these homozygotes for the loss of function alleles can actually end up dying out. And what happens there is these homozygotes, you actually can get rid of certain bad alleles from a population by inbreeding. However, there are a lot of bad alleles to go around. So homozygosity at one locus can actually be ameliorated by heterozygosity at another. So if an individual has two genes and one allele for each one of those genes, if they get both of those alleles, they'll be blind. But one of those alleles, they won't be blind because the other allele isn't homozygous. That's going to actually prevent them from going blind. However, if both alleles are homozygous, that can lead to blindness. This is kind of one of those, some genes can cover for others, and I want you to go all the way back to gene duplication and realize that, if we have two genes for the same trait, if one of them goes bad, the other one can cover for it. Yeah, pretty much. And that's until both are homozygous recessive, and suddenly they're both loss of function, and that's going to be a problem with inbreeding. So there are additional on and on effects where this inbreeding depression can't simply be canceled out in a generation of some lower fecundity, higher mortality individuals. And that's where we're going to see this over time in populations where the inbreeding coefficient, as it goes up, is going to increase the problems that organisms have. So here we have some flowers, and what you see is the coefficient of inbreeding depression. If it's about 0, then you see these traits are not so bad, but as the coefficient of inbreeding depression goes up, so we also see problems with your size of the plants at the spring, size of flowering, size of reproductive shoots. These problems go up, and the deleterious alleles accumulate as they are more inbred. With these birds with an inbreeding coefficient of 1/64, the number of eggs that fail to hatch is about 1/2 of 1 is going to fail to hatch. However, if you have these birds having each 2 eggs, around 1/4 of an inbreeding coefficient, you're going to see, which is, remember, that flowers in the attic case, you're going to see a number of eggs that fail to hatch be about 2. So perhaps a whole clutch failing in 1 year just because these birds are so inbred that there are so many deleterious alleles, the eggs don't even hatch. And we calculate inbreeding depression as delta. Delta is equal to W sub S over O or O over S? S over O. Okay. W sub S over, it's 1 minus W sub S over W sub O. So delta equals 1 minus W sub S over W sub O. S is the frequency of fitness of self progeny, and WO is the fitness of outcross progeny. So the organisms that have been self, so we take this cat and made it to its sister, and we're going to look at the fitness of the kittens that come from that horrific cross. If the fitness there is lower than the fitness of your standard outcrossed cat, your standard run-of-the-mill barn tabby, then we're going to be able to calculate fitnesses and take 1 minus that number right there and get delta. So if these are equal, if there is no penalty for selfing, where WS is equal to WO, then we see that 1 minus 1 is 0 and the inbreeding depression is 0. However, as WS approaches 0 and the fitness of self-progeny goes down, we see that delta is going to increase as 1 minus a number very close to, a very large number, 0 over, 0 over some larger number. It's going to increase the inbreeding depression. So what are some genetic causes of lower fitness in selfed organisms? We can think about different alleles and different genes that may cause lower fitness. And then we're going to look at some of these extreme cases. So selfing, this is when organisms are going to just reproduce exclusively with themselves. Here we see a bunch of plants that are being forced to self. So this is a corn breeding experiment. And what's going on here is you put bags over it's a wheat breeding experiment. You put bags over the wheat tops, and the pollen from the wheat then has to go to those self-same stigmas. Selfing in nature can guarantee fitness without finding mates. What's happening here is this breeding program is trying to keep pure lines and is actually going to be doing something much more complicated we'll come out with later, which is measuring the distance between certain alleles and measuring, QTLs. We'll come back to that with selfing later, but I want to introduce you to that idea that organisms can also self by their own choice. Mating with self can lead to the more rapid rise of homozygotes, however, which is kind of a counter effect to being able to always find a mate. Yeah, but some of your offspring are going to be lower quality. You might not see that until you see actual environmental stress, which is going to bring out these changes in fitness. Now I want you to think back to that nematode example a few lectures back where you have the nematodes that are accumulating mutations. They're fine until they're put in the environment, and suddenly all of those deleterious mutations become truly deleterious. In much the same way, all of these homozygotes for loss of function alleles, they're not really that bad until they're put out in nature, and then suddenly they're lethal. So environmental stresses are often used to purge these poor alleles. We'll come back to that with natural selection. I'll just keep connecting this to the previous lectures and the end lectures. I'm going to review and preview. All right. Let's think about ecology. So the ecology of inbreeding is going to have to do with conservation and endangered species. A problem we see with ecology is that if you reduce an organism's habitat, you're going to reduce their population size. And as you do, they enter what's called the Extinction Vortex. The Extinction Vortex, as you see here, starts with a small population size due to habitat loss and leads to a low tolerance of environmental and demographic stochasticity, i.e. randomness. And also with that small population size is going to lead to genetic drift and inbreeding. So a small population, remember, has a low tolerance to environmental stochasticity. If you have a species of squirrels that requires one tree per squirrel, and some of those trees fall down in a windstorm, well, now you have less squirrels available because you've eliminated some of their habitat, and that small change in a windstorm has an effect on the population size. 5 trees fall down in a population of 500 squirrels, met. 5 trees fall down in a population of 6 squirrels, problem. So this is going to lead to higher genetic drift and inbreeding, which leads to low genetic variation, leading to lower fitness due to, as we saw earlier, inbreeding depression. And since there's a loss of alleles, it leads to low adaptive potential, which makes these organisms less able to tolerate environmental stochasticity, which causes their population size to go down more, which makes more inbreeding, which makes less genetic variation, which means lower fitness, which means lower potential, which means extinction. This is a problem we see with endangered species. Well, is there hope? Yes. Conservation genetics here. So this is a good example of the Florida puma. So this organism, it had a real inbreeding problem where a lot of the organisms were homozygous for many alleles because they were so inbred. The Florida puma had a very small habitat where you have this necessary, essential panther habitat, which is being crisscrossed by highways, which is going to be seen with towns coming up along the highways and people hunting the panthers, and just a big problem. So these endangered species had a real bad inbreeding problem. Well, what they took is some pumas from Texas and moved them over to Florida. We can see here you have your pure Everglades panthers in 1986. And in 1986, there were very few of them. Your pure Florida panthers, there were very few of them. But in your pure Texas pumas were introduced, I can't remember exactly what time, it looks like mid-1990s. They introduced 8 Texas pumas. And those were outcrossing pretty quickly as they outcrossed with the Florida and Everglades panthers to make these blue ones, which have these panthers descended from crosses between your local and the Texas ones. And then you have some of unknown descent, of course, as well. Those pure Everglade panthers and the pure Florida panthers end up kind of going down because you're introducing genetic variability from the Texas pumas. So the introduction of new genetic material was actually able to help conserve these genetically inbred Florida panthers. So there we see where conservation genetics through outcrossing can ensure there a higher amount of genetic variability. So yay, there is a way to avoid the extinction vortex and to avoid the problems associated with inbreeding. Now this is all very genetics and very high in the sky. Let's actually look next time on how organisms assortatively mate and choose their partners. And we'll take care of that in the next lecture.